This conference will now be recorded.
Okay, so good evening, folks. Today, Friday, we are looking at November 5th, this muscular system study guide that I've posted for you all in documents and resources for our class in Unit 4. Okay, so documents and resources, Unit 4, and know that we'll be discussing this evening. I have posted, there's images as well as notes in this, and I think that it's a very concise guide for a good portion of what we want to talk about as far as for the muscular system and such. And really know that with the muscular system, skeletal muscle contraction is a big part of what we're going to be discussing. And I've already shared with you my previous YouTube video regarding the muscular system, and you've seen how I've also embedded video regarding what takes place in those 3 videos regarding skeletal muscle contraction. And so tonight, this is going to be like a review. I know that you've been studying and reviewing things, and I hope that this will make it even a little bit more clear to you all. So know that really when we're talking about muscle cells, when we talk about each individual types of tissue and such, so we're looking at muscular tissue, and muscular tissue is comprised of muscle cells. Well, in the case of skeletal muscle, we're in particular, we're looking at these muscle cells, also known as AKA muscle fibers, right? And so we have muscle fibers in cardiac muscle and smooth muscle also. They're unique, and we'll talk about those in a moment. But know that muscle fibers are also known as muscle cells. So tissue, muscular tissue, is comprised of muscular cells, okay? So let's look at, as far as you should know the difference between the different types of muscular tissue, skeletal, cardiac, and smooth muscle. We know that skeletal muscle is a voluntary muscle, cardiac and smooth muscle are involuntary. We know that skeletal muscle and cardiac muscle have striations present, and smooth muscle, hence the term smooth, does not have striations present. Smooth muscle would appear to be more of a spindle-shaped type of cell, muscle fiber, muscle cell. And so really when we're looking as far as histologically, we're looking at those cells in the tissue slides in the lab. We noticed that we didn't really see the spindle shape of the muscle cell, but we did see the elongated nucleus to represent the smooth muscle. Whereas in the case of skeletal muscle and cardiac muscle has branching. They both are striated. And skeletal muscle, it looks like the fibers are right on top of another, just like you would stack a bunch of logs or something on top of each other. Same thing with skeletal muscle. The cardiac muscle, there's branching present, and you should recall that also. There's these intercalated discs that look a little bit darker than the regular striations of skeletal and cardiac muscle. Now, as we move on here, these are very important terms for you to know as far as excitability, contractility, extensibility, and elasticity. Okay, so excitability, we kind of get this because we realize that, hey, as far as skeletal muscle is concerned, in order for it to do its job, there needs to be an initial electrical impulse or an action potential. So this bioelectricity needs to be present in order to what? Stimulate skeletal muscle in order to begin the process of skeletal muscle contraction. As far as contractility, not only does skeletal muscle and other muscles also, but we're focusing on skeletal muscle right now, not only does the skeletal muscle, is it excitable, but it can also be contract as a result of being adequately stimulated. And you'll see here as far as extensibility, so skeletal muscle, right, can extend or stretch beyond their resting strength and then return back as far as with the elasticity, have a recoil, resuming the resting length after stretching. So this extensibility and elasticity work together, right? And so very important that these 4 terms, you should know what they represent and mean, okay? As far as the function of muscular tissue is concerned, yes, it can produce movement. It can help to maintain posture and body position. As I'm sitting here in front of you lecturing, right, I'm not just kind of slumped over, right? So I have some type of erect posture. I'm not standing, I'm sitting, but I'm still maintaining some type of posture. So my musculature is not fully contracted. And so not all of the muscle fibers are contracting, but a portion of those muscle fibers are contracting in the postural muscles of my body in order to keep me upright. There's also stabilizing joints, and as a result of exercise, as a result of contraction, we produce heat. Okay, and that's a very important aspect of humans, that we do this, as well as other mammals. Now, I'm going to take a moment and I'm going to skip this one for a moment here, but come right to this image. So this image is a great image, okay? And as you look at this here, there's a few things that you need to know, okay? One, that how does skeletal muscle anchor to bone? It anchors to bone via these tendons, right? So tendon is comprised of dense, regular connective tissue. Dense, regular connective tissue. The tendon will anchor a muscle, right? Tendon to the bone, okay? And it will do that on both ends, okay? And know that there are connective tissue coverings present in muscle, and muscle really acts as it's in layers, okay? So what you'll see here, folks, is that this represents the full muscle, right? So the full muscle, that's it right here. The muscle is comprised of these structures right here. Here's one of them, this big structure right here. See how we have this circle around this large structure, which would be one of these big structures. So this whole area, this whole area, this whole area, this whole area, they are called fascicles. So muscle is comprised of fascicles, many, okay? In this case, we're viewing, here's 1, 2, 3, 4, and 5, right? 6, 7, 8, 9, 10. Wait, let me get back for a moment here. See, here's 3, 4, 5, 6, 7, 8, 9, 10, and we're not viewing another one right here behind this, so it'd probably be 11 fascicles comprise this muscle belly, right? So there's the full muscle. So 11, we're saying fascicles. Now the fascicle is comprised of, see these structures right here? That's this structure right here. That's a muscle fiber, also known as a muscle cell. So in this case, this one fascicle is comprised of, let's count them here. So we have 1, 2, 3, 4, 7, 8, 19. We're seeing 19, maybe like 21, 22 is really how many muscle fibers, muscle cells comprise this fascicle. So again, the muscle belly is comprised of these fascicles, and one fascicle is comprised of many muscle fibers. Now, here we get to the next level. So one muscle fiber is composed of many myofibrils. See all these other little dots? And you'll see here mitochondria present also. These are the mitochondria, right? So many myofibrils, I'm not even going to count that, right? But one muscle fiber, one muscle cell is comprised of many myofibrils. And know this, folks, that the myofibril is composed of myofilaments. So the myofibril is composed of many myofilaments, and that would be actin, thin myofilament, and myosin, a thick myofilament. And let's just see here as far as, yeah, so here you're looking at here, this is a muscle fiber, a muscle cell. It's comprised of many myofibrils. These myofibrils are composed of many, and you'll see the dots in here and such, myofilaments, actin and myosin. Understood? So that's an important concept. And then what I do next is then, so I'm showing you how the muscle is made up of fascicles. Fascicles are made up of, comprised of muscle fibers, myofibrils, muscle fibers, also known as muscle cells, okay? And then the muscle cells, muscle fibers, are composed of many myofibrils. And the myofibrils are composed of many myofilaments, actin and myosin, okay? So are there any questions with that first before I move on? Okay.
You had mentioned, oh,
go ahead, go for it.
My question was, are we going to need to draw that out?
No, you will not need to draw it out, but you should know the spelling. Yes, you should know the spelling, and know that again, the myofibrils are composed of myofilaments. Yeah. Okay.
Okay.
And the myofilaments are actin and myosin. Now, the next step is that No problem. And then the next step is that we go back and we see here these terms, epimysium, perimysium, and endomysium, okay? So you should know those three terms. They are the connective tissue coverings, epimysium, perimysium, endomysium. So the epimysium covers the muscle. The perimysium covers the fascicles. The endomysium covers the muscle fibers, also known as the muscle cells. Okay, so I'll say that again. The epimysium covers the muscle. The perimysium covers the fascicles. The endomysium covers the myofilaments. No, covers the muscle fibers, also known as the muscle cells. So the endomysium covers the muscle fibers, also known as the muscle cells. Sorry about that. And then you'll see here where it says sarcolemma. What is that? Well, that's the plasma membrane of a muscle fiber, also known as a muscle cell, right? Because think about this. So the sarcolemma is the plasma membrane of a muscle fiber, aka muscle cell. Okay. Any questions with that? All right, so really important that this is the very basics as far as muscle structure is concerned, skeletal muscle. We're looking at connective tissue coverings. We're looking at the basics as far as how muscle is comprised of. So the way I tell students to think about it is that imagine if you had many markers and you put a group of these markers together. Well, then you can think of all of those markers all together, all next to each other, like holding a bunch of markers, as that would be the muscle, okay? Then take it down to the next level. Imagine all those markers that you're holding together are actually comprised of the muscle fibers that comprise a fascicle. And then it goes on further, right? A muscle fiber is composed of myofibrils, and then myofibrils are composed of myofilaments. So do you see how these cylindrical structures are really the structural foundation for muscle, for skeletal muscle? Cylindrical structures are the structural foundation for skeletal muscle. Does that help when you're thinking about the structure of muscle?
Can you just repeat what you just said about it one more time?
Sure. So understand that when we're talking about skeletal muscle structure, it's composed of cylindrical structures, right? It's like a cylinder. So think about this, right? You've got the muscle here. This is a muscle belly. Say it's the biceps brachii. And the biceps brachii is composed of many fascicles, right? Fascicles. These fascicles are covered in perimysium. The whole muscle itself covered in epimysium, but the fascicles are covered in this dark line here, perimysium. It's a connective tissue covering. This fascicle is comprised of many muscle fibers. The muscle fibers are covered in endomysium, that connective tissue covering. Understood? And then deep within, right, directly surrounding the muscle fiber, when you think of a cell has a plasma membrane. Well, the plasma membrane for muscle fiber is the sarcolemma. And then the muscle fiber is composed of many, see all these little cylindrical structures? They are myofibrils. And then if we were to look at the myofibril, it has many little cylindrical structures, and they are myofilaments, actin and myosin. Actin are thin, myosin are thick myofilaments. I hope that helps. Okay, this is something too that it just takes a while to get it. You have to kind of review it and talk it out and listen to it and go over it again and again and look at other images online can be very helpful also. Okay, so it's important that you have to think about this a bit. All right, so now know that here we have now, and I've given you, so here are the terms that I've actually just written it out for you. Epimysium, epimysium is covering the muscle. Muscle is made up of fascicles. Fascicles are covered by the perimysium. Then the fascicles are comprised of muscle fibers. The muscle fibers are covered in endomysium. And the plasma membrane of a muscle fiber, the sarcolemma. Okay? Now let me just backtrack for a moment here and just see if I'm, because I'll come back to the, so I'll come back to the sarcomere in a little while, okay? But that's when we're talking about actin and myosin, okay? And I think what I want to do, let's see, okay, yeah, let's go over these terms here as far as, so yeah, so the insertion and the origin, okay? So the origin is the anchor. So the muscle, right, connects to bone via tendon. So at the origin and at the insertion, there's tendons connecting to bone, right? Now, the immovable bone that the insertion moves toward when the muscle contracts, so think about biceps brachii. Flexion, that's the action. Extension would be triceps brachii. Biceps brachii, triceps brachii. So for this flexion action of the biceps brachii, is my shoulder moving? My shoulder's not moving, is it, right? So that's the origin. The insertion would be in the forearm. Do you understand? The forearm is where, when a muscle contracts, this part is of, it's really of, sorry, this part of the muscle, the movable bone moves toward the muscle's origin. Do you see that? All right, so origin and insertion, very important. And when you hear the term action, A-C-T-I-O-N, the action of a muscle, it's just when a muscle contracts, and we would say this is called an isotonic contraction, okay? Isotonic contraction. This is when the muscle belly actually shortens. It gets smaller. So do this. Actually, everybody like contract your biceps brachii and you'll feel how it goes from extended, long, to when it contracts, it shortens, okay? And that's a type of isotonic contraction. So isotonic is I-S-O-T-O-N-I-C, isotonic contraction. And we're going to talk about isotonic and isometric a little bit, okay? So we will discuss that. If not this lecture, next lecture, okay? Now you'll see here, let me just check my email for a moment just to see if anybody else had, okay. I'm just keeping my phone on for the email just to see if anybody needed to contact me during class as far as students. So the term sarcolemma, we talked about this, right? So a muscle fiber, also known as a muscle cell, is covered in the plasma membrane, right? The plasma membrane of a cell is what really keeps the cell together. So the plasma membrane for muscle fiber, muscle cells, sarcolemma. The sarcolemma, now let's go back for a moment here. So realize that, right? So the actual muscle fiber, right, muscle cell, it's covered, right? It has the sarcolemma, that's its plasma membrane, but what is it covered in? It's covered in the endomysium. That's the connective tissue that keeps it all together and covers it. But the actual plasma membrane of the muscle fiber, sarcolemma. So don't get endomysium and sarcolemma confused, okay? The sarcolemma is a plasma membrane. The endomysium is just connective tissue. So epimysium, perimysium, endomysium, connective tissue coverings. Okay, let's see here. And so the cytoplasm of a muscle cell, right? So and again, you're going to see this, folks, I'm sorry, but you're going to see this in other tissues and such that will use different names for a name that you already know. So a basic cell, right? Eukaryotic cell, we're talking about the cytoplasm. It's the guts of the cell. So we have the cytosol, the liquid portion of the cell, and all the different organelles and such that are present within the cell, right? Covered in this plasma membrane, right? Including the nucleus and nucleoplasm and such. The cytoplasm of a muscle cell, muscle fiber, is known as the sarcoplasm. And this is going to come important because we'll see sarcolemma, sarcoplasm, and then we're going to see another term called Sarcoplasmic Reticulum, the SR. And we'll take a look at that in just one moment. You'll see here that a muscle fiber is comprised of hundreds to thousands of rod-like structures, cylindrical type structures, right, that are called myofibrils. And the myofibrils are comprised of the myofilaments. Now, so let's look at this here for a moment. So as we're looking at this structure, right? So when we look at under the microscope and we're looking at skeletal muscle and cardiac muscle, we're seeing that there are striations present. Well, these striations are present because there are different myofilaments that are overlapping. Okay, so look at this here, folks. So see the blue? The blue represents the thick myofilaments. So what would that be? The blue would be the myosin. The thin myofilaments, see represented in red, thin myofilaments, that would be actin. Now, this is just a very crude representation, but just helping you to think about this for a moment as far as what's taking place. So when we look at this here, folks, we're looking at that when there's an overlap, there's going to be a darker representation, right, than when we're seeing a lighter representation, right? There's only one fiber that's there's only one myofilament that's present, okay? So thin myofilaments, thick and thin myofilaments, thin myofilaments, and then this line here, this really dark line, that's called the Z line, okay? And that's really, when we're thinking of the functional unit, the contractile unit of skeletal muscle, it's the sarcomere. And this is at a very microscopic level that we're seeing these myofilaments, okay? Actin and myosin. And so think about this for a moment. When you have an isotonic contraction and my muscles will not only just start getting tight, but then they'll shorten, what's taking place, folks, is that the myosin is stable, not moving, but the actin, see these thin red lines? The actin will slide upon myosin, the thick filaments. So the thick filaments do not move, but the thin, well, there's movement, but it's at like there's a pivotal arm that's moving, but it's not the actual filament itself moving. They're stable. But the thin myofilaments, they're moving, they're sliding, and as they slide, that's when you're going to have a shortening of the muscle belly. Okay? So let me ask you let me ask if there's any questions regarding that so far. Because it's a little bit more kind of a strange type of a thing, but if you've watched the videos, and I would tell you to watch those skeletal muscle contraction videos many, many times, folks, okay? Many times, because it's just going to help you to review it and go over it, and I would take notes as you do this, so that this way you're actually writing down step by step what's taking place as far as skeletal muscle contraction is concerned from all three videos, okay? From the action potential all the way to cross bridge formation. And as cross bridge formation, it's just a matter of that. There are a connection between these myosin has those heads like a golf club. They touch with the actin, the thin myofilaments. And we'll look at, let's see if we can look at here we go. So this is where we have that cross bridge formation occur. So here would represent, this is myosin. Myosin does not move other than these structures right here. Okay, the myosin head will actually move, and they're going to actually move and slide actin, this whole green structure here, that structure is actin, that will slide as we have pivoting of the myosin heads moving. Connecting, sliding, disconnect, connecting, sliding, disconnect, right? We've seen this in the video, okay? So now let's look at this as far as at a little bit more of an detailed level as far as what's taking place at these myofilament areas here, as far as, again, myosin and actin. Well, on actin, right? On actin, we have this tropomyosin. See this line here? The line, right? That's actually called tropomyosin. And in the case right now, in this image, it's not blocking the myosin binding sites on actin. It's not blocking them, because you'll see here that there's a connection between the myosin heads and actin. But if it were, right, if this line were blocking, okay, then there would be no way that myosin could connect with actin, all right? Now, do you recall what goes on and what takes place as far as when what allows for this myosin, this tropomyosin, this line here, to move out of position. Well, troponin needs to interact with calcium. So there's a calcium binding site on troponin. Troponin will interact with calcium, and as a result of it interacting with calcium, it will then change shape and move tropomyosin, exposing the myosin binding sites on actin and allowing then for myosin heads to connect with the actin and begin the whole process of contraction. And know this, folks, right here. So you're not seeing, it's a static image, so you're not seeing movement, right? So what I would tell you is that in just this snapshot right here, the type of contraction that you're seeing is called an isometric contraction. Well, what's the big deal? Well, an isometric contraction is different from an isotonic contraction. So when you all think of a contraction, or a muscle contraction, you're thinking of this type of a contraction, an isotonic contraction. I'm contracting and relaxing biceps brachii and triceps brachii, okay? They're antagonistic muscles. Their actions are opposite to each other, okay? They're not working together, they're working in opposition to each other. When I contract biceps brachii, I have to relax triceps brachii. When I contract triceps brachii, I'm relaxing biceps brachii. They're antagonistic, okay? If they were working together, doing the same action, we would say that they are synergistic. They're working together, right? Synergy. But the case is that they're working opposite to each other. So getting back to isometric and isotonic contraction. So in isometric contraction, we have the cross bridges are formed. Now, so think about this for a moment. So I'm going to contract my biceps brachii, but I'm not going to make it move. I'm not going to cause the origin, the insertion to come closer to the origin, but I'm still going to contract the muscle. So see, I've not moved it, but this muscle is hard. It's contracted. We would call that an isometric contraction. So how about your stomach muscles? You can contract your stomach muscles and not do a crunch. I can just stand here, I can sit here, and I can contract the rectus abdominis muscle, right? And it can become hard, contracted, and what's happening is that cross bridges have formed, but there's no sliding of actin upon myosin. So in isometric contraction, there's cross bridge formation, but there's no sliding of actin upon myosin. So I'm going to stop for a moment. I want to ask if there are any questions, or do we get that? Do we understand that? Just give me somebody say something, if you don't mind, just to give me some feedback.
Yes.
Okay, all right. So, and again, you need to watch those videos, folks, because it's very important to review and see and actually watch the videos, because they're so good. They're short, and yet they're chock full of all kinds of good information. Now, another important factor to bring to you here is that where does this calcium come from that will interact with troponin, allowing it to change shape and move tropomyosin away from the myosin binding sites on actin. It's important that you remember that. So tropomyosin will move and expose the myosin binding sites on actin, allowing for that cross bridge, this cross bridge to occur. Okay. And I think I just lost my train of thought with something after I said that. So I'm sorry. All right. Do the best I can with that. I apologize, but that's important for you to have an understanding. So isometric, isotonic contraction. Okay, now let's see here. Yeah, so here we also, this is important for you to see too. So the sarcomere, I said it's that contractile unit of the skeletal muscle, and know that we have these different bands and disc and zone. Okay, and this is just a matter of looking at, and that's why I provided this image for you, so they actually see that the sarcomere, and there are, I don't know, thousands, millions of sarcomeres present in each skeletal muscle. And it's just a section this section from Z line to Z line, that's the sarcomere. And so you'll have the actin and the myosin myofilaments, and whether they're overlapping, causing darker areas, or there's only a single myofilament, there's the lighter areas, okay? And you'll see here where the I band represents, where the A band represents, and here's the H zone right in through in here, okay? Simple enough, not difficult, okay? But you should know this, okay? And let's see here. Yeah, so yep, so let's go look at this here. So again, the myofilaments, actin and myosin, right? The filaments that comprise the myofibrils, right? Because myofilaments are the smaller structures present within the myofibrils. And remember, muscle fiber is composed of myofibrils, which are composed of myofilaments. And you'll see here, as far as myosin is the thick, actin is the thin, actin is what moves, actin is what slides. The cross bridges, they form after the thin and thick filaments link together during muscle contraction. There's that linkage, that's a cross bridge. Tropomyosin and troponin. So troponin, right, major protein in a thin filament, troponin binds to calcium ions, and then what we'll do is that will change shape, and it will cause tropomyosin to move out of position and expose those myosin binding sites, okay? Very important. Now, here's another protein, titin, right? This extends from the Z disc to the thick filament and then runs within the thick filament to attach to the M line. So it's thinner. Titin is a thinner protein present within the whole sarcomere, okay? Now, look at this here, and this is cool. So this would be the sarcolemma, the plasma membrane of a muscle fiber, okay? And so what's going on, folks, is that we have the sarcolemma present, and you'll see here, here's the sarcoplasmic reticulum in blue, okay? Sarcoplasmic reticulum in blue. Now, this would represent one myofibril, okay, one myofibril. And the muscle fiber, the sarcolemma, is the plasma membrane of a muscle fiber. The muscle fiber is composed of many myofibrils. Here's one myofibril. The myofibril is composed of there's many myofilaments in through in here, okay? And so what you're seeing here is that we have this sarcoplasmic reticulum in blue. This contains the calcium. Here is the terminal cisternae in blue, the portion of the sarcoplasmic reticulum. And then the T-tubule is this structure here, which allows for the actual action potential, the bioelectricity, to pass between and inside of the muscle fiber. And what's going on is that once that electricity passes here, there are voltage-gated calcium channels that will open and allow for then the calcium present within these sarcoplasmic reticulum, and in particular, this terminal cisternae of the sarcoplasmic reticulum, to release the calcium. And when that calcium is released, it's going to interact with the troponin and will allow for then tropomyosin to move out of the way, exposing the myosin binding sites on actin. Okay. So really important for you to know step by step as far as, we have, so let's take a look here. And I'll make this a little bit bigger. And so here's the neuromuscular junction, right? We have in yellow here represented the nerve, and we have in red and below and inferior to it, the muscle. We would call this area here the motor end plate, where there's not a touching, so the nerve and the muscle do not touch. It's called a chemical synapse, because this chemical, right, these synaptic vesicles containing the chemical, it's a neurotransmitter called acetylcholine, will be released into this space, and that acetylcholine will interact with specific receptors on the motor end plate of different types of channels that will open and allow for sodium ions to pass through and potassium ions to leave. And so this movement of ions will create an electric potential that will then carry this electricity, this action potential that's produced throughout the sarcolemma and the T-tubule, which will then interact with the calcium channel, right? These calcium voltage channels that will allow for then calcium from the sarcoplasmic reticulum to be released and interact with, and you're seeing here, the here's myosin and here's actin, and interact with the troponin on actin and allow for cross bridges to occur. I'll stop for a moment. Any questions with this information so far?
No, I'm good on this one.
Good, excellent. All right, so then let's move away from there. Let's continue on here. So that sarcoplasmic reticulum, it's also known as just the SR, okay? It's now know this, right? It's a smooth ER, sarcoplasmic reticulum, which contains the calcium, right? So calcium is important. So look at this for a moment here, right? So calcium is important when in this region right here of the nerve, because this is the motor neuron, this is the axon terminal, the end of the motor neuron. When that action potential drives down and through in here, what does it do? It will activate calcium channels, voltage gated calcium channels that will allow for calcium to enter in to the axon terminal. Recall that? So calcium enters into the axon terminal here, which will allow then for the synaptic vesicles here containing acetylcholine to release, they'll release the acetylcholine into this space, this synaptic cleft, which will then allow for the whole process of muscle contraction to begin. So calcium is important here, and it's important in here, being released from the SR into the area where we have the myofilaments, actin and myosin. Okay. And
Dr. Perrone, are you gonna have this particular picture posted on the documents and resources?
Yes, I will post this for you, indeed.
Thank you so much.
Oh yeah, sure, no problem. Absolutely. So to get back there, so calcium is important in two areas, two times for skeletal muscle contraction to occur. Okay. And you'll see here that really the bottom line, and I've highlighted here and put it in bold, sarcoplasmic reticulum holds on to calcium. What does the skeletal system do? It's a reservoir for calcium, is it not, right? So sarcoplasmic reticulum, a reservoir for calcium, regulates intracellular levels of ionic calcium, and it stores calcium and releases it on demand, right? So very important. We looked at already the terminal cisternae and the T-tubules. The T-tubules will be conductors for the action potential, the electricity. The terminal cisternae are just a portion of the sarcoplasmic reticulum, and they are just an expansion of and will release the calcium into the area of the myofilaments. And just know that this term triad just represents that there are two terminal cisternae and one T-tubule, so that's three structures known as a triad. Okay. So when all of this that I'm talking about as far as skeletal muscle contraction is concerned is the sliding filament model of contraction, meaning that what slides? The actin myofilament slides upon the thick myofilament myosin. Okay. All right. The term action potential, the electrical current that needs to be generated in the sarcolemma, in the plasma membrane of a muscle fiber, for that muscle fiber to contract. Okay, so really important. I would tell you, I would make flashcards of these terms throughout this study guide. I would also really take my time, and I've given you the steps here for skeletal muscle contraction here, and in more detail, so what's going on as far as what's going on with the motor neuron. And this right here at the end, so this is important as far as getting and understanding what's taking place as far as the generation and propagation of an action potential. So I have to tell you that this whole electricity type thing and understanding about what's going on as far as the plasma membrane is concerned is important for neurology also. So it's a very a foundational important principle with neurology. So we look at muscles contraction and see what's going on as far as this action potential is passing down this motor neuron, right? This motor neuron ends up here, opens up those calcium voltage gated channels, calcium enters in, right? You see how the action potential is important here. There's also an action potential that is going to be produced here, right? So we have electrical event, chemical event, electrical event. Do you see that? Electrical event on the nerve, chemical event in the synaptic cleft in that space, and then an electrical event will occur again here at the motor end plate and then be passed throughout the plasma membrane of the muscle fiber and enter deep into the muscle fiber via the T-tubule. Okay. So excuse me one moment. Let me just, okay. So let's do this. I'm gonna go up here. Yeah, okay. All right, so again, contraction. I said to you that there are two types of contraction. Isometric, where we have cross bridges form, but there's no sliding, isometric. Isotonic, that's when we get the idea of shortening of muscle, the muscle fiber, the muscle itself. That's an isotonic contraction. Isometric, cross bridges. Isotonic, shortening of the muscle fiber, shortening of the muscle itself, not the muscle fibers, but the shortening of the muscle itself, okay? Again, it's known as the sliding filament theory. Sliding filament, the actin slides upon myosin. That electricity, that action potential is really important. And again, it's important at the nerve, and it's also important on the muscle. So it's electrical event, chemical event, electrical event. And know that when we say that the neuromuscular junction, the nerve muscle junction, is an chemical synapse. A synapse just means that there's an actual communication between two different structures. Okay. Now, there can be electrical synapses like we have present in the brain and such, where we actually have direct connection. Okay. But in the case of the neuromuscular junction, it's a chemical synapse. So it's the chemical that passes between the nerve and the muscle to allow for communication. And I'm going to give you two other terms that you should know. It'll help now, and it'll help also in neurology. So the motor neuron with the axon terminal, we would say that this is presynaptic, P-R-E, pre-synaptic, because this is the synapse right here. I mean, we understand that it's the synaptic cleft, it's that space, but really that's the synapse. This is the synapse here. This is where we have communication between the nerve and the muscle. They don't touch, okay? There's that gap here. So that's the synapse. So presynaptic, and then postsynaptic would be the muscle, the motor end plate here. That would be postsynaptic. So presynaptic, post, P-O-S-T, postsynaptic. That's going to help us in next semester when we're also talking about, in particular, where we're talking about different types of situations where it's not only just the neuromuscular junction, but we're talking about the autonomic nervous system, and this is the somatic nervous system, as far as we're looking at muscle contraction taking place and the interaction between the nerve and the muscle. We're going to be talking about the autonomic nervous system and looking at what's taking place with sympathetic and parasympathetic responses, and those types of neurons are different and such. And so we'll look at that in more detail next semester. But presynaptic, postsynaptic, important for you to know. Okay. So I'll share that with you all. Okay. Now you'll see here as far as steps for a skeletal muscle fiber to contract. So let's look at this here. The muscle fiber is stimulated by a nerve ending so that a change in membrane potential occurs on that motor end plate. There's an action potential that's produced in the sarcolemma. Sarcolemma, again, is the plasma membrane of the skeletal muscle. The action potential, that bioelectricity, automatically is propagated along the sarcolemma, that plasma membrane of the muscle fiber. The intracellular calcium ion levels rise briefly, providing the final trigger for contraction. And so that's really important that we have then the increase in and the release of that calcium ions into the space where we have an interaction between the myofilaments and such, and so that there's then the allowing for cross bridge formation to occur. Again, I would highly recommend that you take this guide and you create flashcards for it, and then you write things out many times. That will be very helpful to you all. Neuromuscular junction, again, that nerve muscle junction, that's the chemical synapse. Again, the neuromuscular junction is a chemical synapse, because what's that chemical? You'll see it right here, acetylcholine. And know that what will happen to acetylcholine? Well, in the first video, you saw that acetylcholine esterase. It's an enzyme. ASE, that's the suffix. Acetylcholine esterase. That will help to break down acetylcholine into acetic acid and choline, right? And then what will happen is that the whole cycle will begin and more acetylcholine will be produced in the axon terminal, okay? The synaptic vesicles are what actually hold onto the acetylcholine. They have that chemical, that neurotransmitter present within, and will release it when calcium is released into the axon terminal. So a motor neuron stimulates the skeletal muscle fiber by, so that nerve impulse, that action potential, reaches the end of an axon, right? It goes down to the axon. The axon terminal releases acetylcholine into the synaptic cleft. Acetylcholine, that's the ACH, diffuses across the cleft and attaches to acetylcholine receptors. And know this, right? Throughout the whole body, you're going to see that there are specific receptors for specific chemicals, okay? We would call those, I'm blanking on the term. It'll come to me, so apologize. But it's important that you see that there are specific receptors for specific chemicals, okay? On the sarcolemma, on the plasma membrane of the muscle fiber, the acetylcholine binding triggers electrical events that ultimately generate an action potential. So again, coming back to this image here, electrical event, chemical event, electrical event again, action potential, release of acetylcholine, the chemical, and then interaction between that acetylcholine and the receptors, which will create then an electrical event here and transmit along the sarcolemma and the T-tubules. And here's that term, acetylcholine esterase. So very important to see that it's an enzyme. You see the ASE, the suffix there, located in the synaptic cleft, breaks down acetylcholine into its building blocks of acetic acid and choline, okay? Terminates in the effects that occur after acetylcholine binds the acetylcholine receptors and prevents continued muscle fiber contraction, right? So this is important to know that. So you're not just walking around in a state of just tonic contraction, right? No, there's contraction and relaxation, and it's can happen quickly, but there's also situations where not all the muscle fibers are contracting at once, depending upon the load. So think about this for a moment, right? So I have my phone here, okay? So lifting up this phone, it's pretty light, right? But I would say maybe this water is a little heavier. So the muscle fibers that contracted to pick up my phone are not as many as the muscle fibers that would contract in order to pick up this bottle of water. Okay, and so that's also something to keep in mind as far as when muscles are contracting, not all of the fibers present within the muscle are contracting. All right, let's see here. Yeah, okay, and I have more information to share with you as far as I'm working on another study guide, okay, to finish up the chapter and such, and I'll share that with you all on Monday, okay? But this is the foundations as far as skeletal muscle contraction are concerned. I want to talk more about ATP that's being produced. I want to talk about specific types of muscle fibers because we have different types of twitch type fibers, slow and fast twitch fibers. So we'll talk about that. We'll just talk about a few more things regarding muscle on next class. So you'll see here that the events in generation and propagation of an action potential, okay? So what's going on now, folks, is that we're talking about on the muscle itself, right? So at the motor end plate of the muscle, this is what's going on. And know that sodium ions are moving, potassium ions are moving, and this movement of these ions will cause a depolarization, will cause a change in the resting membrane potential of the plasma membrane of the muscle fiber, meaning the sarcolemma. And that depolarization will then allow for then events to occur to begin to produce that action potential, okay? So this is something that takes a little bit to, it's not an easy concept, and it's a little bit more like, huh? But again, remember, and I tried to introduce this right from the very beginning, the movement of ions changes what takes place as far as that resting membrane potential of the plasma membrane, whether it's nerve or whether it's muscle, okay? Remember, a resting membrane potential, -70 millivolts, okay? And that changes, and I'm gonna be sharing with you more video for the next chapter in neurology that will help to give a little bit more of an understanding regarding how these action potentials are being produced. Just right now, know some of these basics here as far as if there's always movement of sodium and potassium ions. Sodium ions are always moving first. Question? Let's see. Okay. All right, anybody? Okay, so Catherine, do you have a question?
No, I'm actually with you on this one.
Okay, so I'm just gonna turn off your mic though, okay? Okay, there we go, thank you, good. All right, so let's hide everyone again, share my screen, good. Okay, good. All right, so you'll see here that as we go to 2, and oh, I gotta just, excuse me folks, one moment, I just gotta tell someone to. My mom and dad are checking on me. See, it doesn't matter how old you are, mom and dad still care about you and want to see how you're doing. So, yeah, right. I know they're 79 years old. They're in very good shape for 79, thank God. But yeah, they're cute. So depolarization then leads to then, if we depolarize, if we change the plasma membrane from positive on the outside and negative on the inside, that's the resting membrane potential. If it's reversed, that's a depolarization event. Returning back to that positive on the outside, negative on the inside of the plasma membrane, that would be then a repolarization, a return to the resting membrane potential. And you'll see here that repolarization is a consequence of changes in the membrane permeability, so that now it's not so much the sodium ions moving, it's more of the potassium ions moving. So I said to you that it's always, remember, sodium ions move first, then potassium ions. And you're just gonna have to like watch the video. The video will really help to see as far as watching the animation and seeing what's taking place as far as ions moving back and forth as a result of channels being opened or if they're closed. Remember that we have leak channels that are always open, and then we have ligand-gated channels. See, now I didn't even see the term, but ligand-gated, a ligand, that would be a chemical. So in the case of acetylcholine, right, that's the chemical that interacts with the specific receptors for acetylcholine, and that will in turn act as the key to open the gate and allow for sodium ions and potassium ions to move. Okay, so I've given you a lot in less than an hour, or roughly an hour or so. There's been a lot of information here we've covered, and let me just see one more. Let me see one more. This is also, I'm going to share this image with you all. I like this. This is a cool image, just showing you step by step what's going on as far as not only, and I'll talk about this in more detail next class, but not only just the cross bridge formation and then the movement of the sliding of actin represented in blue here, upon myosin represented in red, right? But also the importance of ATP. And that's why I said next class talking about the generation of ATP and why it's important. And know that with ATP, ATP allows for then there to be a separation of a release of the cross bridge. If there's no ATP being generated, being synthesized, being produced, then we end up having the case of in a cadaver, we would say it's stiff. Why is it stiff? There's muscle contraction that's taking place and it's stuck. There's no ATP present in order to release the myosin heads from the actin. And so we have this sustained contraction, the sustained cross bridge formation, which would be what then? It would be, we would say it would be in the case of like an isometric contraction. The muscles are there, so they're not like cranking on and moving, although there have been movement of bodies and such, like flexion of the torso a bit and such. So there have been some strange things, but really primarily it's just an isometric type of contraction where we have cross bridge formation not being released because ATP isn't being produced to allow for the release of the cross bridge. And we would say then that the cadaver is stiff. And we call that darn, say it's tough when you're on
Rigor
medication.
mortis?
Pardon me? Say again?
Rigor mortis?
Thank you. Very good.
You're welcome.
Yep. Awesome.
Hey, I have a question for you.
Yeah, go for it, please. That's why I'm here.
Is that theory with the actin and everything like that you had just mentioned?
Yes.
Like I had a reaction to compazine where I had dyskinesia and my muscles locked up. Is that the same idea?
Yeah, so really, as far as I can't tell you the whole, I'm sorry that I'm not in pharmacology wise. I don't know all what's going on as far as the
Oh yeah, but
I apologize. So I don't know. I could look that up, but I don't know off the top of my head. I apologize. But
yeah, I'm just wondering, like dyskinesia.
yeah, so when you're having issues where either you're not able to fully control muscles, or they're moving and they're kind of spastic or not, and it's different. And I'd have to look up the pathophysiology. I don't know. I'm sorry.
All right, I'll scope it out. No problem.
Yeah, cool. And if you want to share with us, that'd be good.
Will do.
Yeah, good, excellent. Anybody else? Okay. All right, folks. Very good. So I'm going to stop recording.